And I will go directly to the second slide. I would like to start with this really nice picture, a cryoEM image of an LNP system. But what can we see here? We can see spherical-like forms. We can see blebs and twins and LNPs, and with all in different shades of gray and varying significantly in their size. This formation seems to stand out from the surrounding by the lipid layer, and this is typical for liposomes and LNPs. So, but what can experts see? The expert can realize what RNA is hidden inside and outside the system. But note, always are only a snapshot and limited to a really small view window. And we should be aware, LNP systems are highly dynamic systems. cryoEM is in fact a really fascinating technique, but it will never be used under GMP in routine control, but could be a really helpful pillar during the development of new technologies. Manufacture of LNPs that provide LNPs only for research purpose. We have to, here in the yellow box stated, the criteria has to be delivered, so meaning size and charge. But if we deal with LNPs used in therapeutics, we have to consider various additional critical quality attributes, etc., potential distribution, or the concentration of the RNA in and outside, and also regarding the integrity. So this is why we developed here in-house a strategy that cover the selected criteria, and currently on the left side, on the left side here only a few techniques are available at the moment, focusing to the intact particle system. So ELS, electrophoretic light scattering, and DLS are well established, but either we create only limited parameters, or we are too imprecise. This is why we consider also Taylor dispersion. This is really new, and CZE of intact LNPs with a special fluor for labeling, but also AF4 could be really helpful tools for the characterization. On the right side, the analysis of the molecular level with the focus of the RNA, and this means we have to learn a lot of new things, poly-A tail of the RNA, or the differentiation between single-strand and double-strand structures, but also the quantification of unencapsulated RNA and the total. And at the end, really important, the correlation between the analytical results and different biological potency tests. This is of a special interest to understand the relevance of the here deduced analytical parameters. Later on, I will show you new results from 3 different LNP systems using a defined analytical package. But before, I will give you an overview of these techniques. From the sober strategy now to colored balls. Using an RNA specific labeling to the intact LNP and combined it with the electrical field, so it's on the left side, when we get the information regarding all RNA containing phases, structures, forms. If I do it, the same procedure with lipid-specific labeling reagents, I get the information regarding the ionic mobility profile of all lipid-containing but also micellar phases with this technique. Considering or looking to not modified LNP systems, DLS is really well established. And here we derived the hydrodynamic radius by a combination of light scattering with Brownian motion. cryoEM in the same category is like a picture book which tells us exciting stories whereby we learn a lot of morphology and structure. And Taylor dispersion is so far here in-house under development. We don't yet have it under control, and we can at the moment not derive any conclusions, but we can use first results to compare different LNP systems, also in the context of all other techniques. And last but not least, the analysis with a focus of the RNA, of the unbound and free RNA, and also with total RNA after degradation of LNP system using two methods, size-based CE and charge-based. So I would like focus on these two techniques, CZE in the combination with specific fluorophore and also Taylor, as these two techniques are really new. And the interesting thing here is we can use both with the same equipment, either applying a pressure or in CE applying a high voltage. In CE, the capillary is filled with a suitable buffer at the defined pH. And if we're using here cyber green as an RNA labeling, we know the mobility of the RNA. This is clear, it's anionic, but we have no idea regarding the mobility or ionic mobility of the lipid particle. So Taylor dispersion is more a mathematical framework for the analysis of dispersion, where it results from the interaction between a parabolic flow and the radial diffusion of an analyte, where it is injected as a plug. When we evaluate the peak variance, and this peak variance can when transferred to a diffusion coefficient and later to a hydrodynamic radius. So I mentioned CE and ionic mobility, but why is ionic mobility so important for particles? I know electrochemistry is not everyone's cup of tea, but the understanding of the zeta potential is really indispensable and important in particle analytic, as relevant for in vivo behavior and relevant for the stability of the dispersion. Really briefly, when the charged particle is dispersed, a double layer, the Stern layer and a diffusion layer, is established on the particle. And within this diffusion layer, there is a hypothetical plane, the slipping plane. And one part of this diffusion layer is stripped off when a particle is migrating in an electrical field. And this slipping plane is the zeta potential. This plane is a characteristic parameter of each particle and is strongly dependent on the pH, ionic strength, and also concentration. At defined conditions, the zeta potential of nanoparticles can be deduced when by the ionic mobility, as shown here in the equation. There are in the moment two possibilities, two technical possibilities. How can I get this ionic mobility. Whereas the electrophoretic light scattering, this needs a really fingertip feeling, patience. You need a high experience and you need a lot of material. For CE, you need only a suitable dye. So now enough with the theory. I start here with the first LNP system. On the left side, the CE profile of a LNP stained with RNA specific labeling. And what can we see here? This profile is really reproducible. But we can see here all RNA containing phases, structures, whatever, in a range between the 0 point, the neutral point, and the anionic range. So far it's not clear which signals are related to which morphology or which structure. Changing the fluorophore to a lipid specific dye on the right side, we obtain a similar information here. The signals are shows at the same migration time. Also signals are when related to the micellar known aqueous phase. And below, both DLS and Taylor dispersion shows clearly the presence of particles. However, it cannot be differentiated if we can see free RNA and the LNPs or both together. And for Taylor, using also a RNA specific labeling, the profile seems to be really reproducible, but we have a significant tailing at the rear edge, and this is a strong indication where adsorption occurs that we have to develop in working on this technique. Summarizing, we can say we have lipid particles, phases containing RNA with different ionic mobility, so meaning with different zeta potentials. And we have particles in the range between 8700 nanometers. This system is highly dispersed. And now, when we injected the intact LNP system without any treatment, we can see on the left side, in the green electropherogram, the size variance of the free not bound RNA. The same for the charge-based technique. And if we disrupt the LNP systems with the surfactants, it's clear we increase the signal when we analyze the total RNA. At the moment, it's quite difficult to say how to interpret the CZE of an intact particle. Experiments in particular, but the TEM image alone shows that it won't be a piece of cake. The next candidate, a completely different system, but in general, the picture is very similar. Both RNA and lipid structure are present in similar ionic mobility ranges, and only the shape and the profile is a little bit different. The lower size distribution evaluated with DLS and Taylor dispersion is striking. In the sum, we have a similar behavior, lower size distribution, and also anionic structures are present. And what's about the RNA? Here, a similar picture. Both CE techniques reveal that RNA is unbounded present. See the green lines in both techniques. And what we also see is the main part is fragmented. And after degradation of the LNP, we already know it, we have a strongly increased of the signal and get when on the left side the size variance of the RNA and on the right side the charge profile. It is really interesting to note that the RNA fragments on the left side are in the same size range. It was really astonishing for us and we have no answer. And the TEM, the picture reminds me on the soap bubbles. but the spheres are to some extent really perfect. So, and the next and last candidate, this is a batch, another batch of LNP2. And this seems to be completely different with regard to the CE experiments. The signals significantly reduced and we have no anionic structures. But what does it mean? Are there LNPs present? Where is the RNA? And the DLS profile clearly shows where something is wrong. And with Taylor dispersion also we have no clear answer, only when it looks really abnormal. So fortunately, we can have a look at the RNA level, at the molecular level. And if we only had with pictures, the world seems to be better again. Not only the encapsulation efficiency, but also the integrity of RNA looks perfect. No fragments of the RNA are present. However, cryoEM reveals the truth. In the sample, oh, sorry. Sorry. So in the cryoEM, we reveal that there are large empty liposomes and the dense material outside here of the LNPs or of the liposomes are aggregated forms of RNA. It looks like that the LNP are completely degraded and converted. And in contrast to the LNPs before, here it was a really fresh material from the manufacturer. At the end, there is still a lot to do, but the first decisive steps have been taken. And the good news is that we can get a first good feeling regarding the presence or absence of healthy desired LNPs without utilization of cryoEM. We can consider some of the methods as completed, and for another part, we have to think again and start it again with new experiments and perhaps also underpin results with potency tests. Professor Huwiler will tell us more about this topic, parameters and potency tests in the afternoon, and I'm looking really forward to this talk. I thank you for your attention and will also thank the team, Christina, Ayub, Sarah, Kirill and Marius, that was involved in this project. Thank you so much.